I'll just repeat what I told. Okay. First thing is, you guys will be taking notes, and we'll give a common topic where you guys have to do a case study or do some study on the topic, and you have to create a PowerPoint presentation, and one person representing your team should present it here in this class. Okay, this is to know about how well you are good at presenting, and how well you are analyzing a scientific problem. Okay, so this is so important in the study. Next thing is, uh, some of you people have enrolled for internship, and for that we'll be telling you the difference between research paper and a review paper, and how to construct a research paper or how to construct a review paper. And people who have enrolled for this internship, we can submit a report based on that. Okay. And next thing is, soon after completing all, like one subtopic or a topic, we'll try to give you a demo or a hands-on training. Okay, a session, 1 hour session. One class will be dedicated for that. And since we have completed NGS data analysis, today I'll just show you how to do fast QC and Trimmomatic. Okay, we'll just see, and just follow the steps, and you guys can try it out. And what you can do is you can take it as an assignment, and you can submit the data in a document wise. Like, you can include the screenshots, you can include the fast QC report as a HTML file, or you can convert that HTML file into PDF file, and you can incorporate that file into a thing. Okay, so this is like you submit a assignment to your class. Okay, we can make it that make that happen, and this will be for easy for me to analyze, or just get to know how better you guys understood the topic. Okay, so am I clear?
Yes, sir.
Yes,
Yes.
sir.
So you guys have any other doubts regarding these three things?
No, sir.
No. So first of all, uh, let me share my screen. Okay. So first thing, you guys are new to Ubuntu. Some of you, at least, you guys are new to Ubuntu. So what I'm gonna do is I'll just show you what are the basic commands. At least I'll just show you top 50 commands that uh, will be using. Okay. So is my screen visible?
Yes, sir.
So
Yes,
these
sir.
are the top 50 Linux commands you should keep in mind, or uh, so uh, in order to work with Linux, because Linux is totally a backend environment. Okay, you will be doing commanding in order to analyze or in order to do some stuff. Okay, so is the screen legible? Like, you guys could see this, all these
Yes, sir. Yes,
commands.
sir.
So yeah,
Yes, sir.
so you have seen a couple of commands yesterday, and now we're going to see about this 50 commands. I just don't want to show you all the commands, okay, to run that, because we don't have much time. I'll just explain. Okay, ls is just to list what is there in the directory, and pwd is present working directory, cd is change directory. You have, when you have to change from one home directory to the next directory, you can use cd. And mkdir is to create a folder or a directory. Okay, mv is to move a particular file or a folder to a different folder. Okay, and cp is for copying file or folder, rm is for deleting or removing a folder or a file from a directory, and touch is to create a empty file. Like likewise, you create a text file, you can create an empty file which has nothing in it. So touch is a command used for it. Okay, and in just okay, and clear, clear is to clear the screen, and cat is for concatenating a document, and you can edit the document to open in terminal. Echo is for provoking a call, and as like if you wanted to provoke an action, or you should provide some privileges, or you wanted to edit something, and bash scripting, you can use echo. Okay, cat and man are pretty much same. You can edit or you can modify editable data. That is maybe a document, a text file like that. Okay, who am I is one of the commonly used command to know what is the configuration of the system or how your system is configured or what is your system is. Okay, and grep is like the search option what we have in Windows. This is used to search something which is hidden in a directory or somewhere around. And it's all not just um, not that common ones. I'll just tell you the common ones. Zip is to compress a folder, and as likewise, unzip is to like unzip a folder. Okay, and SSH is where you provoke or you call a cloud-based system into your terminal to use that system in your local system. Okay, and services, you can see what are the services running behind, like Apache browsers, and we'll be using Tor like that. Okay, some services will be running behind the terminal. You can just check for that. And kill all are what you will be doing in task manager, right? You'll be ending the task if it is not responsible. It is not responding, right? In that case, you can kill the task, okay, what is running. And chmod is where you give full privilege to configuration file. Ifconfig is where you identify or you can check on the network configuration, whether you have connected to Ethernet or you connected to any WLAN, that is Wi-Fi or something like that. And wget is a command that basically you can just use to download from a link or from a website. Okay, that can be of any file type. So we are going to use this command. And apt pacman yum rpm or package types, you can just for updating packages or for calling packages, you can use this four commands. Sudo is the administrative privilege we'll be using, and where is where you find where command, like where is particular file or where is a particular folder. And if you wanted to know the property of a file or a folder, you can use what is command. And if you wanted to add a user with the administrative privilege, you can use this user add. And if you wanted to create a password for the administrator, you can just click on password. Okay. So these are the most common ones. I didn't explain all, but I'm just explaining the most common ones was what we are going to use. Okay. So you have any doubts with this? I'll share you a link so that you can just go through it later on this commands, especially. Just a minute. So I'll just put this link in a in the chat box. You couldn't hear anything? Am I audible now? I was speaking.
Yes, sir.
So I guess people
Yes,
are
sir.
having some network issues. So yeah, okay.
Sir, repeat once.
Sorry, you guys were telling something? Yeah, it's audible now. So yeah, that's great. So now I'll share my screen once again, and we'll just see the step-by-step process on how to download a SRA file, that is a fastq file from the open web, and how we can run fastqc in terminal. And if it is there, if it is having adapter sequence, and we'll see how to trim the adapter sequence, and we'll run this fast QC once again, so that you'll be completing this quality check for a file. Okay, so that particular thing is what we're going to do today. Okay, so, So, so I guess my screen is visible now. Uh, where's that? So, this is the homepage of SRA from NCBI. SRA, I told you already. SRA is Sequence Read Archive. Okay. From here, you can just download the FASTQ files or SRA files. Okay, if you're downloading SRA files, you can use SRA toolkit. It is also one of the command line tool to convert SRA files into FASTQ files. Okay, so I have a list of list of files. Okay, list of files with their accession number and database where it can be taken. Is this visible? I'll just zoom it out. Okay, so
Yes.
if I'm going to give you these accession numbers for your hands-on, okay, and you'll be trying it out, and you should mention in your report that what kind of data you have taken, what accession number, and once you go into the SRA database, there'll be a general description, so you can include those descriptions in the file too. So this much data I have, like accession numbers I have. I'll be sharing you this file. Okay, so for now, what I'm gonna do is I'll just search for this particular accession number here in SRA. Okay, I'll just give search. So it is the RNA sequence data of Aedes albopictus, and it's a mosquito. It is done on mosquito cell lines and for Zika virus. Okay, okay. So here I have searched for it and you got this got the data. You can include all these data. This makes sense. Okay. So first you can take this, you can read what it says about. Okay. And from here you can click on this run accession number here. I guess you guys could see this here and click on it. It'll be opening. It'll be opening in a of- a different window. And here from here you can just click on fast and fast queue download. Okay,
So can you
and
repeat the step once?
once again, yeah, okay. So
Your
what I did
slide is
is
not visible.
my screen is not visible for all.
It is visible, sir.
Yeah, I guess you guys will be having some network issues. Okay, so I'll just repeat what I did. Okay, so this is the homepage of SRA in NCBI. Okay, so as of now I've taken the first example, that is the Human Genome Project, and its RNA seq data. I'll just copy it and I'll paste it here. Okay, and I'll search for it. Okay, so it is the RNA-seq data which is run on Illumina MI-seq machine, and it is mostly used on transcriptomics perspective. Like when we do transcriptomics, this is the data what we gonna further analyze. So when I search here, I'll be getting a page like this. Okay, so there are a lot of disease and there are a lot of organisms. So apart from this list, what I have, you can also search for other things. Okay, so you can also search for other datas like NGS, E. coli. Just give a keyword like this so that it'll give you the possible results. Okay, so you got a DNA sequence of E. coli and NGS of some libraries. Okay, like this you'll be getting. So the most um like likely one, or the most uh the correct one will be on the first. Just click on it and you'll be getting the accession number there. You got the accession number. So this is the accession number, or from here you can just click on and you can download it. So you can see it is a large file. Okay, so the size is around 11.5 GB. Okay, so For this study alone, we are going to focus on some small sized data. Okay, some small sized data. So likewise, I have downloaded a data already. Okay, it is there in my downloads. So you guys could see here this one SRR 293834. This is one of the file I downloaded because this is the smallest file I could find. Okay, to make this like easy, I just downloaded this file. Okay, I have downloaded by clicking on here, by clicking on let it load. Okay, so here I clicked on this run accession number, and from here I clicked on this faster and fast queue, and you can download any data, any sort of data from here. So if the particular data is huge, it prompts that the run exceeds the download limit more than 5 gigabase pairs. So in this case, what you can do is you can use SRI toolkit to download. Okay, if the sequence is lesser or lesser than 5 kilobase pairs, you can see the kilobase pairs here. It is 39.1 G bases, so we can't download it directly. We can use SRA toolkit to download this data. So previously we seen a data which there was an option to download it, right? So these are two methods. If the total base number is high, the total number of bases in the file is so high, you can't download it directly from the website. What you could do is you can just use SRA toolkit to download. SRA toolkit is also one of the toolkit collection of tools uh, that you can work in terminal. Okay, I'll just show you how it works. And if the bases are less than uh, yeah 5 G bases, you can just download it directly from here. Okay, by clicking on this, you can just click FASTQ file and it will be downloading. So it shows download here. You can download this kind of data, this FASTQ file to your local system. Okay, so able to follow till now?
Yes, sir.
Okay, so you know
Sir.
how to download the file. So clicking on its accession number, you'll be prompted to this web page. From here, you can click on to fasta/fastq download, and when you click on it, you can click here fastq to download the file. Okay, so now it will be there in your downloads folder. Okay, so what you could do is you can just copy this file, and once if you successfully installed your WSL, that is Windows Subsystem for Linux, it'll be showing you here in the hierarchy. And here you can click on Ubuntu, and your folder, it'll be always there inside home. Click on home, and this is my administrative folder. Inside this, I'll be having all the data. What you can do is you can just paste it here. Okay, so I already pasted it here. So I'll not be pasting it again. So this is one of the approach. So this is the direct GUI based approach, because this is just a subsystem of Linux. You can directly just locate the folders or the directories of Ubuntu itself from Windows like this. Okay. So what I did, I just went to download section, download folder. I copied the thing what I downloaded, and I searched for Ubuntu subsystems. I went to home. I clicked on my administrative or the administrator folder. Here, click on that administrative folder and I just pasted it here. Okay, so clear till now?
Yes, sir.
So, so this is one of the method. Other method is what you could do is I have installed SRA toolkit already. So I'll just yesterday, since I told you I'll be giving the command and you could copy and paste it there and you can try it out. I told like that. So I'm cooking that document. So I have given those steps in that document. I'll be sharing it to you across once we are done with this class. So I have this SRA toolkit already. Okay. So in that there is an option to dump the data or to download the data. Okay. So that option is fastq hyphen dump and space. And I wanted to dump this particular accession number in the sense I'll be giving that accession number. Okay. I'll just giving the accession number alone. This is the accession number. Okay. So I just copied it. What I'm going to do is I'll just delete. Okay. RM and I'll delete this file from this because I'm going to show how to download using SRA toolkit. Enter. So RM is the command used to remove any file so that when you list it out, that file will be missing here. Okay. So what I'm going to do is fastq c fastq hyphen dump space and this accession number and enter. So it'll be downloading it. Okay. It may take some time. Okay. Just wait for it. So it have downloaded the file here. So when I click ls to list the directory,
Sir,
you can see,
उसको
yes,
repeat, sir, repeat once, please, sir.
yeah, I'll repeat it once more. Okay. Yeah. So SRI toolkit is collection of tools one of the collection of tools here in terminal in Ubuntu terminal where you can just download when the file is larger in size using SRA toolkit and the command is fast hyphen dump. Okay, so when you know the accession number, so when you know this accession number alone, what you could do is instead of clicking and downloading it from here, I showed you another method like the locating the WSL directory and pasting this file, the downloaded file there in the Ubuntu directory. So that is one of the approach. It will be easy for you guys, okay? Since you're working in WSL. So when you have a whole Linux system, only Linux distro which is working on your computer, on that case, this won't be working because that is not a subsystem. We can't locate it directly. What you could do is you can just use this terminal to download the file. Okay, so this is my current working directory. So I'll just check it with pwd. Pwd is the present working directory. I'll be knowing on what directory I'm currently in. So I'm here in home and SAM directory. Okay, so I just wanted to list what is there, what are the folders and files is there in the SAM directory. I'll give ls and it shows me what all the data we have here. Okay, so what I did, I downloaded this particular fastq file with the help of fastq-dump tool, which is present in SRA toolkit. Okay, I've installed the SRA toolkit already. If you wanted to know how I install, I've given the steps in the document which I'm preparing. I'll share it across. So uh, I have downloaded this particular file using fast-dump method. Okay, so I'll show it again. rm and I'll delete this file. Like I'll delete this file. So I've deleted the file. It is not there in the home directory. You can see it is not there. So what I'm going to do is I'm going to use this fastq-dump method to download it again. If you know the accession number, you can just type fastq-dump and give the accession number. It will be downloading the data for you. Okay, so till now it's clear.
Yes, sir.
Yeah.
Yes, sir.
So would you guys be able to replicate this in your system? so far?
Yes, sir.
Yeah. So it got downloaded in the home directory itself. Let me show you that ls for listing the directory, and it is there in the SAM, that is my administrator folder itself. Okay. And yesterday I created a folder called test where there are files there to run fastqc. So this is just a test folder. It's there. Okay. What I'm going to do is first I should check the quality of this. Okay, since I've downloaded directly from the web, I should check the quality of this data. What I'm going to do is you are familiar with fastqc now. So type fastqc space and this. Or else what I'll do is I'll just copy this uh, file to test directory or test folder, so that all the data which is generated, the data which is generated from fastqc will not be there in my desktop. That is this SAM folder. Okay. So to do that, what I'm going to do is I'll just type cp, and this is my present working directory, right? I'll copy this, paste. Sorry. This is my present working directory. This is how you locate to a directory in terminal. Okay, SAM, and this is the file. Okay, so CP is copy this particular file from this directory to space. You have to give a space to the same thing until SAM. It's fine. To there is a folder called test here. You can see test. Okay, I'll just paste test here. T-S-T test. Okay, and I'll click enter. So what it does is it copies this file and it makes a copy on this test folder. So to view that, what I'll do is I'll just change the directory using cd command space test. Okay, so I've changed the directory to test, which is there in the administrative folder, and I'll list it ls. So you can see this fastq file is already here. Okay, so these are the sample files which I run already. So now we're going to run this particular file, SRR 29383404, the 409. Okay. So we downloaded this using fastq dump, or else you download it directly from web and you locate it in the uh, downloads folder. You copy it and you can paste it there in the WSL, your Ubuntu directories. Okay, like this. Or else you can just dump using this fastq dump using the accession number. So these are the two methods to download the SRA or fastq file. Okay. So soon after doing that, it is there in my desktop. Well, let's consider this SAM folder as my desktop. So it's there in my desktop. Okay. I am making a copy of this file to a folder called test, which I created already. So this test folder is where I run this fast QC analysis. So inside this thing is there. This fast Q file is there. Okay, I'll clear the window now. So you guys able to follow till now?
Yes, sir.
Okay, so let me list this. So I have this file. What I'm going to do is fastqc space this file name, and I'll click on enter. We'll be doing the analysis. Okay, since the size of the folder, this file is around 34 MB, so this have completed soon. So I'll list the directory again, and you could see it have given me a HTML file. Okay, and a zip file of it, a compressed file where you have these icons, images, and other data associated with this HTML file. Okay, so to view this, what I'm going to do is I'll come to my home, that is the Windows thing, and on Ubuntu, sorry, on Ubuntu, I'll click on home and I'll click on the desktop, that is SAM, and I'll click on test. Okay, so when I open it here, you can see the FastQC result is present here in the HTML format. So I'll just open it. I'll double click. When I open it, it'll be opening in a in a HTML format in your system. Okay, so we can see when it got run, like when this analysis happened, and what is the file name here. And these are the common informations, basic statistics, statistical information. And we already seen about this pair based quality sequence quality and per tile sequence quality. So this is having lot of uh, ugly bases, or we can call ugly tiles, because this red color denotes the high level of error or high level of contamination, we can say like that. And you will be having a report like this. Okay, so we got a report right now. What I'm gonna do is, so this thing we didn't see that day, right? It's the overrepresented sequence. So these are adapter sequences which is present there in the file. Okay. And what could be the possible sources is given here, true seek adapter index one. So these can be the adapter sequence files. These are called this configuration file which is having this adapter sequences. So these are the adapter sequences, and you can see the adapter content. So all these will be in a graphical data. What you could do is you can just view this in a HTML format, or you can jump into the folder that is your Ubuntu, and you can unzip this folder, like unzip space, copy the file name and enter. So what it does is it unzips the file. It decompresses the file. So when I click ls, it gives me a folder like this. So you can see a folder like this. It unzipped and gave me a file. The zipped folder, the compressed folder will also be there, and also the decompressed folder will be there. So to check that, I'll just go there to the test directory, refresh it. You can see the folder here. I could open it now. You can see all the data. So we'll be having a summary in the form of text format, what it has done. So it has passed the basic statistics. It has passed the per base quality sequence quality run, and it has failed this per tile sequence quality. I've given a picture for this per tile sequence quality in the slide deck which I shared you. So that is the clear one, and this is the failed data. Okay, it has lot of tile sequences which is not in a good quality. Okay, that is what this particular picture mentions, and it gives you pretty much the summary of whether the test or the parameters passed or failed. Okay, and you can take the image files from this. It'll be having all the graphical image files, the graph image files or the spectrum image files. Okay, like this, you can just use this data or this file in your uh, research paper. Okay, here this folder will be having these kind of data. Underscore data is a text document where you have the log of of what the run had done to this data. Okay, so these are informations which you get accordingly to the graph. Okay, this is a graph. You want to split it into a comma separated value or Excel kind of data. For that, we can have this particular fastq underscore data file, and you can convert this into a Excel sheet. Okay. Yeah, someone raised their hand. Just a minute. Yes. Tell me, Toshani, what's their doubt? You raised your hand? Yes.
Hello. Am I audible?
Yes.
Sir, I wanted to ask that when you were giving the copying command, that you were copying the file from SAM to test folder. After that step, you have given a CD. I think that change directory command. Sir,
Yes.
I was not able to get it. Like if we have already copied the file, why are we using the CD command here?
cd command is to view or to change to the directory what we are currently working on. Okay, so let me show you here. So sorry, I have cleared the screen. So cp is to copy a file. Okay, so for example, I'm just going back to the current folder pwd. So this is the home slash sam. This is my desktop. So when I view it, when I list it, I could see these are the folders and file which is there in my desktop. Okay, cp is a command used to copy a file and put it to another folder. Okay, so I should go and check whether the file is copied to the folder. So to do that, I'm just changing the directory and going to the folder test. So now I'm currently at test folder, not in the SAM folder. Can get it now? I've copied the file, but I wanted to check whether the file copied is there in the test folder. So I just changed the directory to test, which is already there. The folder is there in there. And on that test folder, to go to the test folder, I just gave cd test, and in that test folder I came here. You can see shows me I'm here currently there in the test folder. You can check it with pwd command on which directory you're currently in, and I'm the test folder, and I copied it there in this test folder. To check it, I'll be giving ls. So the file is here. CP is for copying and CD is for changing the directory and moving to the different directory. Likewise, I'm here in this folder. Okay, like let me say I'm here in this SAM folder. The SAM folder consists of multiple folders and files. Okay, I copy the file. I just paste it here in the test folder. I just wanted to check whether this file is there in the test folder. So I'll double click on it and I'll change the directory to test folder, right? This is what I done there in terminal. Here,
Okay, sir. Okay, sir. Got it.
yeah, yes. And anyone, does anyone have any other questions? Yeah, Vashini, tell me.
Sir, you said we have to use the SRA toolkit for this, right?
Yep.
So you have provided the installation steps in the document, right? So like the other document you are talking about.
Which I'm preparing right now. Yeah, I have.
Okay, so we have to install the SRA toolkit and then do all that process which you just explained, right, sir?
Yes, if the data what you're downloading is more in size, it is more than 5 G basis, then you can rely on SRA toolkit to download, and you can copy the file and paste it there in the your Ubuntu terminal. Or else, if the size of the file is lesser, you can do what I did earlier. I'll just go there to the here. I'll be clicking on the accession number, and I'll click on faster and fast queue, and I'll download it here. Clicking this fast queue button. Okay, this is done when uh, it is there in the file size or the total bases are lesser. The total size of the file is 4.5 MB. Okay, so the total bases present in this particular file is 552 megabases. It's not gigabases, right? So if the total basis is greater than 5 gigabases, then you have to rely on SRI toolkit to download the FASTQ file. When this is lesser than 5 gigabases, you can directly download it from this website itself by clicking on this FASTQ button. Like clicking the SRA accession number, it will be landing here in this page, and you can click on FASTQ and you can download it. Okay.
Okay, sir, got it. Thank you, sir.
Yeah, thank you. So, uh, where I enough? Yeah. So, I've got the result. Okay. I've got the result, and I came to know that by seeing the result, there are some, and we'll just focus on this particular adapter content alone. Okay. Others, we'll just leave it here. We have this. This is a failed parameter. This is failed because it is overrepresented sequence. There should be no overrepresented sequence or any adapter sequence in the file what you have. So if this is there, we should eliminate it using Trimmomatic. Trimmomatic is a tool, what I told you. It is used to remove the adapter sequences. okay, or any extra sequences present in there. So you can see it is around this will be around 50 base pairs in sizes. Okay, so these are overrepresented sequences. We have three different sequences. Okay, and you can take this in account through seek adapter, index 3, index 2, and index 1. You can see you have to take this in account, this word in account. While I'll tell you, this is a most significant word. When I run Thermomatic, you'll understand. Okay, so uh, the thing is we have overrepresented regions, and we also have warning on adapter content. We also have warning on adapter content. So we're going to rectify this. Okay, you can see the color of the line, what it represents. It represent universal adapter, Illumina universal adapter. So this particular NGS data was produced by a Illumina a high-seq software and a high-seq machine, and there they used universal adapter for this run. This to sequence this, they used universal adapter. That is why it is given in red color. So what I'm going to do is I'll just switch to the directory. So I'll just switch to the directory, and here I'll be running this command to run Trimmatic. So Trimmatic command is a huge one. So I've run it already. I'm just searching for it. So this is the command. Okay, this is the command. So I'll explain you. Trimmatic is a Java-based tool. So that is why we are just calling for Java, and we are just switching. We are giving an argument to run a jar file. Okay, it's a jar file present in the Trimmatic folder. For easy understanding, what I'll do is I'll just copy this and put it there in the chat box. So this is the command to run Trimmatic. Okay. So before doing that, what I have to do is we should relocate to the Trimmatic directory. So cd space double dot will take you to the previous directory, and ls here we have this Trimmatic cd. Please don't mind the noise behind. Okay, and enter. And I'll click on ls, and you can see it has a jar file. That particular folder has jar file, and there is a folder called adapters and a license. So there are different types of adapters we'll be using in the next generation sequencing based on the type of sequencing. So cd adapters ls. So these are the adapter files in the form of FASTA file format. So each FASTA file format will be having segments or different types of adapters present in it. Okay, it is like a reference file to check for the adapters which is there in the uh, FASTQ file which we have downloaded, and to eliminate it. Okay, so these are the adapter FASTA files. We're gonna compare our file, our FASTQ file, with these adapter files, adapter FASTA files, and if it is there, we'll be eliminating it. Okay, so I'll just move on to the previous folder. Sorry guys, please don't mind the noise. Okay, uh, so would you able to follow till now?
Yes, sir.
Yes, sir.
Okay, so Thermomatic has a folder called adapter, where it has the several adapter sequences of different types based on the type what we are used for running the sequence. There are adapters what we use, and those sequences will be there in this FASTA file format. Here the adapter folder of Thermomatic. Okay, we are gonna use these adapter sequences and keeping this as a reference, we'll be searching for the adapters which is there in our downloaded FASTQ file, and we'll be trimming it off. Okay. So to do that, I'll copy, or else we can just run the command here itself. So you can see this command. I've copy pasted the command there in the chat box. You can use that. Okay. So since this Trimmomatic is a Java based software, I'm just calling Java, and this argument is telling me to take the Java file, this jar file, to run this jar file which is there in this folder. And SC is single end. I'm telling this single end. Single end is where you run a sequence and you provide a data only from one end to the other end. Usually, what they'll do is they'll run the sequence, they'll run this sequencing for forward and reverse. Okay. Using adapter, and the sequence will be having both forward sequence and a reverse sequence. And sometimes in some RNA sequencing cases, and we discussed about this RNA sequencing yesterday, right? In some RNA sequencing cases, the RNA data will be here in single end. That is, the sequencing will be done on one end, like wow, A to B. Okay, that from one end to the other end, not to the reverse. Okay, so and we discussed about this Fred scores. So this file comes under Fred 33. There are Fred 33 quality score and Fred 64th quality score. We'll discuss about what this Fred quality base score tells about later. And I've given this, I'm telling this is a SC file, and it comes under this Fred 33 quality score. And I'm telling the input file. I'm just giving the input file, this file. And I wanted a output file like output underscore trim.fastq, trim.fastq. And I'm giving a command like Illumina clip. I have to clip and trim the sequence using this trueseek3 sc.fa. You can see it here, seek3 sc. You can see here this file. I'm giving this adapter sequence reference file here in order to compare it with the input file what I'm giving. And I'm asking this tool to give it give the trimmed data as a output underscore trim.fastq file. Okay, these are other options which I'm giving. That is, I need to refer this particular file, that is trueseek3 sc.fa. Okay, and I'm giving an argument of leading 3, trailing 3, and you guys could see this, the command. And minline 36. Uh, these are the default parameters. These are the default parameters. After this Illumina clip, right? These are the default parameters. Only thing is we should know what kind of FASTA reference file, FASTA adapter reference file we should run. For identifying this FASTA reference file, you can just click on to the FastQC report. You can see TrueSeek adapter. We can see TrueSeek adapter of index 3. So it says here what kind of adapter they have used, what are the possible sources of the adapter, what they have used, and this particular run sequence should be present in the file. Okay, so by keeping this in mind, what I'm doing is I'm giving a command that to cross verify my data, my FASTQ data, with the adapter TrueSeek3 hyphen SE FASTA adapter reference file, cross verify it and trim the adapter sequence. This is the command which I'm giving. Okay. And I'll press enter and it'll run. Okay. It'll be taking some time because it cross verifies with the TrueSeq3-SE uh, file, this adapter's reference file, and it'll be snipping of the adapter sequences.
Could you repeat the steps for the Trimomatic tool you said, sir?
Yes, I'll repeat it. Okay. So this is running now. Once it's done, I'll repeat it again. Okay.
Okay, sir.
What I did is this is the command what I'm going to use. It is not a easy command. I know this is not an easy command. I like fastqc. Thing is, since this is a Java oriented tool. This is built on Java. I should run Java, invoke Java first, and I'm giving an argument -jar. That is, this argument says that take this Trimmomatic -0.39.jr jar file to run this jar file. To specify this jar file, I'm giving an argument called -jar, and I'm telling that this is a single end sequence data. That is, it has only sequenced from one end to other end. It is not having two-sided data, pair end data. That is from A to B or from B to A. Okay, so this SE switch tells about it is a single end data where the sequence is done only on one direction. Okay, and I'm giving the input file. I wanted to trim this input file. So this is what the file we have, right? 409. And I want the output file to be named as output underscore trim dot fastq. Okay. And next thing is I'll be calling a tool within Trimmomatic. A method within Trimmomatic is called Illumina clip because this is a Illumina based sequencing data because this is a sequence data produced by a Illumina machine. So I'll be using Illumina clip. And since this is the updated reference file, I'll be giving TrueSeq3 hyphen SE. SE is single end. This is one direction file. So I'm giving se.fa. And these are uh, these leading three and trailing three and window. Just a minute. It's not running. Right? Just a minute, guys. I guess this is the command. Sorry, yeah. The command was wrong actually. It got an indent here. Indent in the sense it got a space in the command so that it couldn't recognize this command which is given earlier. This command.
Sir,
So
also
I
what there was triple C in that command.
this one, this triple C.
Yes, sir. This
No, I
triple
didn't
C
give.
stands for what?
Okay. Is this there in the chat box too? No, it is not there in the chat box. This is the correct command. The thing is, what I did was killed I killed this process using Control C by pressing Control and C. That is why it is showing multiple C's at the end. Okay. And why this command didn't work? It has got a indent or a space here. So this is the sliding window 4 is to 15 and minimum length of 36. This is the common parameters should be in this format, but I gave this in this format. The data I passed the command in this format. That is why it didn't run. So you can see started the arguments. It has started and automatically using 4 threads. It'll be giving this data. And when you list it, you could see whether it got run. No such directory. Okay, we encountered an error here. What we'll do is we'll just run it once again. PD. I'll move to the uh yeah. So the problem is we don't have the file here. You guys could see the file should be there within this Trimmomatic directory, but the file is not there. So we encountered the error. So we'll just make a copy there. Okay. CP. So before that, I'll just take pwd. This is my home directory. Copy. CP. To uh, so do you guys able to follow what I'm doing?
No, sir.
Can be tricky,
Can you
but
please
I'll just
repeat
explain.
what you did just now?
Yeah, I repeat it. Okay. So what I did is when I run this, it encountered an error. Okay. It says no such file or directory. Okay. So what would be the problem? When I listed the Dramatic directory, it has only adapter sequence folder and the jar file which is running. But I didn't specify what file should be run, what file should be run. Like I gave it here. Is it breaking? Is it breaking?
Yes, sir. Your
Yes,
Yes,
voice
sir,
is.
it is breaking.
Yes, sir.
So
Screen is stuck also, sir.
just a minute. I guess I oh, sorry, I exceeded my data package. Just a minute, guys. Can you guys hear me now? Am I audible?
Yes, sir.
Yes.
Yes, sir.
Okay, okay. So my data package has expired, so I've just connected to another network. Okay, so we'll just complete it soon. What I did is I passed the command. Okay, I specified the file, but this file was not there in the directory on the Trimmomatic directory. Here you can see I moved on to this Trimmomatic folder and I passed this command. But previously on this Trimmomatic folder, the file was not there. The FastQ file which I wanted to run was not there. So what I did is now I'm copying the file which is there in the home folder or the desktop here to the Trimmomatic folder. So this is the command to copy this SRA file which is there in the home folder to the Trimmomatic folder. Okay, I'll click enter.
So there's some space there, right? So
Yeah, it's a
maybe
space. Yeah, it don't work, I guess. Thank you. Rectify the problem. Yes. And yeah, it got copied. So ls. So we have a copy here. And when we go to this Trimmomatic, move to this Trimmomatic folder, we also will be having the same thing. So ls. See, now we can see we have the file here. So we'll run the command again. So the command is here. Click. And yeah, so the problem was the file was not there in the Trimmomatic folder already. I copied it to the Trimmomatic folder, and now the Trimmomatic is successfully completed. Okay, this message should be there. And this is a log which it gives. Okay, the input read was 17,177, surveying around 17,073 was the original one. and it eliminated 104 base pairs. That is 0.61 percentage of the sequence got eliminated. This is what it says. So you should be getting this completed successfully message. Okay. So I'll click on ls. You could see the output trimmed FASTQ file is there. Could see this. So could you guys able to follow till now? Yes. If you guys have any trouble in understanding, please let me know. Yes, no, maybe. Any answers?
Sir, when we'll practice by ourselves, we'll come to know how much we have understood.
Okay, so once
Sir,
you have
but
to
we are
do it,
able to follow you. Sorry.
yeah, that's great. That's great. Thank you, Surinder. And Harshia, that's true.
Yes, sir, because now you
We
are
are
telling
having
us we are
this.
understanding, but when we will try by ourselves, we might get stuck in
You'll be
between.
encountering different problems, right? Yeah, that is true. That is true. So okay. Anyway, the session is recorded. We'll share you the session also. So far, it is successful. And what we'll do is we'll just run this fast QC, or else uh, yeah, we'll run this fast QC for this output file where we'll check whether the data got modified or not. You see. So I told you yesterday, since this is the quality checking step, it can be done on each and every alternate steps. Like first step, you can just check for the base quality of the sequence, and after trimming, you can check for the quality, and after aligning, you can check for the quality like that. So ls, I've got a HTML file here. I'll go and view that HTML file there in the folder. I'll Sam Trimmomatic, and this is the output trim files data. So now this has not got eliminated. The thing is, we have used TrueSeek 3 single adapters, right? That could be a problem. So what we can do is we can just try with this another folder, another file here. We can try with different file I showed you earlier, right? So we have different set of FASTA reference files. I used TrueSeek 3-se.fa. So it doesn't do any much changes to the data. It didn't trim the adapter sequence when it compares to the original one. This is the first original one. Okay. But I've got the same data here on the output. It didn't got eliminated. So what would be the problem? I have to change my approach. What I have to do is I should be try running the files. I should be try running the command with other TrueSeek files. TrueSeek files. Okay. We have three set of TrueSeek files. And since this is a single end, we have another single end file called trueseek2-se.fa. So instead of trying this trueseek3, I'll once again run with trueseek2-se.fa. Okay. So till now you got you understood the thing. Yes. So it is more than 1.5 hours. Like it's already 1.5 hours. Okay. So yeah, we'll directly jump into the query section, or we'll just discuss. Okay. Just discuss for a couple of minutes. Like, and we'll wind up in another 5 minutes. Is that okay?
Yes, sir.
No, if you have any questions, please ask me. We'll make it quick. So you understood the workflow, right? You all understood the workflow. I'll just explain it again. Uh, the thing is, uh, I have the data of, I have the accession number. I searched in NCBI SRA web. Yeah. this NCBI resource, SRA resource. I navigated, I clicked on the SRA accession number. It prompted to this page, and from here I clicked on FastQ to download the FastQ file. So when the bases are lesser in size or the file size is lesser, I can just click on to FastQ. Yes, someone have question? Yeah, Varshini, tell me.
So could you summarize the Trimomatic steps like in
Summarize. Okay, I'll tell you. Okay, I'll just summarize overall. Okay,
okay,
is
sir.
that fine? Yeah. So where did I
Yes.
leave? I clicked on this fast queue and I downloaded it, and it was there in my downloads folder. From here, from here, I copied it and I relocated to the Linux WSL working directory at home, and under this the desktop I pasted it here. So this is one of the approach to copy a folder and to paste a folder and to navigate from Windows to Linux. This is the direct approach. We can do like this. When you are using a full-time Linux machine, what you could do is you can copy it directly and paste it there. Okay, this can be done. Or else you can rely the help of SRA toolkit. You can install SRA toolkit in your Linux, and with the command called fastq dump, and you can give the accession number. If you give this accession number here, it will be downloading it. Okay. This is the command you can use to download the FASTQ file within the terminal. And when the FASTQ file is larger in size, you can use this FASTQ dump thing command. Okay. Next thing is what I did. I was copying it and copying the file, and I was pasting it in Trimbomatic folder to make a copy. And first thing I did analysis, the first primary analysis of FastQC on the downloaded file, and it gave me a, HTML file. So we viewed that HTML file. This is the HTML file. So we just focus on this Trimbomatic first. So for that, we have to take this overrepresented sequence and adapter content primarily. And from this data, we came to know that this is the Illumina universal adapter is present more in this sequence. So uh, while downloading it itself, it shows data from uh, it is the data from a Illumina machine. Okay. So what I did, I came to Ubuntu, the terminal, and I made a copy of this file to the Trimbomatic folder, and I passed this command. This command, it is just Trimbomatic is a Java-based tool, so I'm just calling Java, and I'm giving this hyphen jar argument to read or to execute the jar file which is present in the Trimbomatic folder. So this Trimbomatic folder, once downloaded and installed, you'll be having adapter folder, different adapters, and a jar file to run. So I'm just specifying that. Sorry. And I'm just assuming this is a single line sequence. Just just a minute. Yeah. So I pass this argument called SE, and I gave a switch called Fred33, and I given this FASTQ file. This is the input file format where I wanted to trim. Yeah. So this is the input file format, SRA29. This is the input file. And I wanted to rename the file as output underscore trim.fastq. And this is where I pass on the parameters. Lemna clip. I just wanted to refer my sequence with trueseek3.fa. So I guess I made a mistake here. I wanted to specify this hyphen SE here. So I didn't specify that. I just commonly gave trueseek3. I should be giving trueseek3 hyphen SE. Okay. And sorry. And the leading is 3, trailing. Let me tell you about this parameters in upcoming classes, or I'll give the definition for this parameters, the document which I'm creating. Okay. So these are the common uh, like default parameters I'm running. And after doing this, I'll be getting a successful message here. If the run is completely completed, and if we got this successful message, you can see it got generated a data on the output file format what we asked for, like output underscore trim.fastq, and you can run fastq for this same file, and you can check whether the Tribomatic has done its magic, whether it got eliminated, the adapter sequence got eliminated. If not, what you're going to do is we're going to specify different reference file here, a different reference file here, to seek the hyphen sc dot fa, or any other file, like where was the file? So those reference files. If uh, do anything, the output data. So I someone's telling, okay, I'll be trying with uh, files, reference files, app folder, I can different app, uh, start trying those here, and putting it output file where this one of adapter will be removed, and it will be passing this pass this parameter, and overrepresented sequence will be removed. You got the idea now? Sorry if I was like running in two speed, guys. You able to follow? Yes, Surinder. You have any questions?
Yes, sir. Yes, sir. I just wanted to confirm that we are using adapters, right? There are multiple forms of adapters. So
Yep.
my question was that do we need sometime need to do a multiple iterations of like the traumatic step with different kind of adapters to get the desired result, or is there any
Yes. The
like
thing is,
was that right?
to know about the adapter, first thing you can know it from the fast QC result. Okay, pretty much you can check on what kind of adapter sequence you should check with. So here it shows the possible source as TrueSeek adapter. TrueSeek adapter is the commonly used adapter in Illumina machines. So if you're giving a data to run and the particular person runs that NGS or does that in next generation sequencing using a Illumina machine, they'll be using Illumina universal adapters. So in this graph, you can note it down. This Illumina adapters are there. You can see this graph. So with this graph, you could know or you can just assume what kind of adapter you could use. Okay, in this adapter folder, adapter folder, we have different types of adapter. P is for pad end. Pad end is on both the direction, like for example, 5 dash to 3 dash and 3 dash to 5 dash. And on SE is also called a single end. Only it has the sequence of only one point to another point. That is one direction. Okay, so since the data which I've downloaded was the SE data. This is the single end data. This is the sequence file of one end. So I specified the file name to TrueSeq3, which is the updated sequence reference file to seek3 TrueSeq3-SC, and I ran it. Okay, I did the analysis. So soon after doing the analysis, even though it gave me a result, but this sequence didn't get eliminated. Okay, so in that case, what I have to do, I have to go for another adapter sequence here, like SEFA. It is the TrueSeq2. Maybe the adapter sequence which is not present in TrueSeq3 will be there in TrueSeq2 -SE. So before doing that, in this, while downloading itself, you'll be able to find it out in the SRA resource page. Okay.
Okay, sir.
So
Thank you.
yeah, see from here, you can check this is a Illumina machine. This is the Illumina machine, and this is the RNA-seq data. So technically, it is the single end. Okay, so I should be passing, or I should be using only these two parameter files, TrueSeq3-SEFA or TrueSeq2-SEFA. Sometimes with this page, in this page, the full information page, it will be giving pair end. And here it will be given as DNA-seq or chip-seq like that. In that case, what you have to do, you should be using this adapters. TrueSeq2 PE, that is pair end FA, or you could be using TrueSeq3 PE.FA. Okay, now it's clear. So the information
Yes, sir.
about the adapters, you can get it from this page, or else you didn't get any sufficient information regarding what adapter you should choose. You can just go and run the first fastqc of the downloaded file, so that in the fastqc file, in the overrepresented sequence region, you'll get a data, like you'll get an information that this is the possible source of thing where they could be using TrueSeq adapters. So TrueSeq adapters, and the machine will be Illumina Universal Adapter. Since this is a product of Illumina machine, this red color line denotes Illumina Universal Adapter. So what you could do is you can just try with these two files. Okay, these two reference files. Fine.
Yes, sir. Thank you.
Yeah, thank you. And does anyone have any other questions? Sorry. Okay, I'll share you a file where all these commands are put, or all these commands are there in the file. You can just copy paste the commands and you can try it out. Okay, I'll share that file across. You please try it out and try to create a report. You can try to create a report in form of that. You can add screenshots of this. You can add screenshots of the terminal, or you can add screenshots of this particular data, the HTML data, and you can also add screenshots of this downloading your file from SRA, or this home page of SRA. You can include screenshots, be creative, and you can just give the steps in the report. And there's nothing called a false report or a false result. Anything what you got is a result, so it's not a problem. If you are encountering an error, please note it down that too. Note that down in the report too. Okay. And if you encounter any other problem, please ping me in WhatsApp. I'll try to help you. Okay, so that if you guys try it now, we'll discuss about both the results and possible variations what you got, or what are the other possible results what you'll be getting, and we'll be discussing on that tomorrow. Okay, is that fine? So now I'll be sharing you the accession different accession numbers, or else you can just search for different accession number for yourself from the SRA data, and you can just download. You can do the approach what I did. You can download directly from SRA by clicking here on the accession number and clicking on fast queue, and clicking on fast queue to download. You can follow this method. Okay, don't need to follow this SRA toolkit method, because you have to install SRA toolkit in your Ubuntu terminal first, and you should configure it. You should be giving this administrative privileges to that. So I guess you guys don't know about it. So tomorrow I'll show you how to give the privileges. So as of now, you follow this method, directly downloading it from web and copying that from the downloads folder and placing it there in the Ubuntu working directory, Ubuntu home, and you're given a username for your uh, terminal, right? And it'll be shown as a folder, and under that folder you can paste it. That is called a desktop. You can paste it there. From there, you can just use the command cp to copy and paste it in different folders. Okay. And soon after copying that file, you can come on to Ubuntu and click on ls to view it, and if it is there, you can just run fastqc on the file, and you can come back to the windows and you can locate to the folder, and you can uh, click on the output file, and you can click on the output file. We'll be having this fastqc report as a HTML file. You can view that, and you can do this Trimomatic. Don't worry, I'll give this command. I'll give the proper command to you. Only thing is, you should know whether the file which you are selecting is a single end. Okay, is a single end. So if it is a single end, you need to give these two files as the reference adapters, or else if it is not a single end, we have to change the parameter. You should change the switch here to PE, and this option to FRET64. Okay, so try downloading a FASTQ file, which is a RNA-seq data file, and which is a single end file like this. Okay, is that good? Is that okay? Able to follow? Like this. Okay, should be layout as single, and it should be a RNA-seq data, and it should be a product of Illumina machine. Okay, so you guys have any other doubt? Yes. Okay, if you guys don't have any other queries, we'll just wind up with the session. Please try it out. Okay, I guess you guys downloaded Ubuntu, and you made it ready. Okay, only thing is I'll share the document. You can copy paste the commands and you can try it out. If you encounter some problem, please update that problem in the WhatsApp group with a screenshot if possible, so that we could rectify what would be the problem or the error. Okay. Yes.
Okay, sir.
So yes, we'll wind up the session now. I don't want to take much of your time. It's already 1 hour 44 minutes. So thank you so much for joining. It's happy learning with you guys. Take care and please try it out. Okay. If you have any queries, you can ping me there in WhatsApp. Yeah.
Thank you, sir.
Thank you, sir.
Thank you. Thank you so much. Thank you all. Thank you. Yo, I'll end the meeting now. Okay. Take care. Bye bye.